The talk that I'm going to give today, I've titled it Cancer's Faustian Bargain. I'll explain what a Faustian bargain is. Exploiting whole genome doubling in solid tumors. And so I want to start off by just talking about kind of the history of chemotherapy, kind of frame the foundation of the story that I'm going to tell. So, the story of chemotherapy begins in the 1940s. And so after World War I and then during World War II, there's a lot of research and studies being done using mustard gas. And what they found is that troops that were exposed to mustard gas experienced both bone marrow and lymph node depletion. And so there were some pharmacologists at Yale named Goodman and Gilman. If you don't know them, these are like the godfathers of pharmacology. The pharmacology textbook is called Goodman and Gilman's. And so they, making this observation, wondered, could this be used to treat lymphoma or leukemia? And so what they did is convinced a doctor and a patient at Yale to try to inject some mustard gas, a derivative of that, as a chemotherapy. And they found that it worked, that the lymphoma receded briefly, before coming back. And what they discovered is that, these mustard gas kind of derivatives are alkylating agents. So what they do is they add an alkyl group to DNA. And what it does is it covalently binds the DNA together. It cross-links the DNA. And so then when the DNA tries to replicate, it introduces a lot of DNA damage, kills the cells. And so there are a lot of chemotherapies we use that work in this way, these alkylating agents, these busulfan, melphalan, bendamustine, cyclophosphamide. So these all work in this way as alkylating agents. Meanwhile, at the same time in Boston, Sidney Farber was working on folate analogs. And so, what he was discovering was that there's this machinery in cells, this factory that makes DNA, and that folate was this key ingredient to being able to do that. And he worked on these analogs that would bind to the enzymes that use folate, but would inhibit them rather than activating them. And what he found is that that would just completely blow up the system, right? And that the cells are no longer able to make DNA and the cells would die. And so, this mechanism, it was of action was kind of termed an anti-metabolite. And what it does is it just inhibits DNA synthesis. So any cell that's trying to divide and make new DNA experiences cell death when treated with one of these. So he was working on methotrexate, which is still used today for lots of purposes. Other anti-metabolites are like 5-fluorouracil, capecitabine, pemetrazine. Moving kind of into the next decade, in the 1950s, pharmaceutical companies started just screening compounds to try and see, what these different compounds did. And so this is a Madagascar periwinkle. And this is like a flower that's used medicinally in lots of different cultures. And so they, were screening compounds from all sorts of plants, including this one. And what they found in this plant was this group of compounds they're called vinca alkaloids. And so common ones are vincristine and vinblastine. These are antimitotic drugs. Okay, so what they do is they bind to microtubules and impair kind of microtubule dynamics. Other common antimitotics are drugs like paclitaxel and docetaxel. And this is an image that I took using a microscope showing a cell that's dividing. And so in green are the microtubules. And so there's no laser pointer, I don't think. But if you look at the things highlighted in green, so all of the cells kind of surrounding the middle one, these are just cells hanging out in interphase. The green microtubules are kind of forming the structure of the cell. If you if you were to treat them with one of these drugs, it's not really going to do much. It's just going to inhibit the microtubule dynamics. But the cell in the middle is a cell going through mitosis. And so you can see all of those green strands are trying to gather together the white chromosomes. They're trying to line them up during mitosis. And so this process requires these microtubules to grow and shrink and move around to be able to kind of move these chromosomes around. And so, if you're a cell that's going through mitosis and you inhibit these dynamics, it becomes very lethal to the cell. The cell will die during mitosis. And so that's the mechanism of how, these chemotherapies work. Moving into the 1970s, based on the earlier work done with those, the alkylating agents, the mustard gas kind of derivatives, DNA crosslinkers were discovered. So these are the platinum-based chemotherapies, cisplatin, carboplatin, oxaliplatin. And this is, they work in almost the exact same way as these alkylating agents. They just don't stick on alkyl groups, but the platinum is what crosslinks the DNA, sticks it together, introduces DNA damage when the cells try to replicate their DNA, causes cell death. Then into the 1980s, there were topoisomerase inhibitors. So topoisomerase you can think of as like kind of the scaffolding that supports DNA during replication. So when DNA opens up and tries to replicate, you need topoisomerase there to kind of stabilize the DNA. And by inhibiting topoisomerase, it causes this disaster during DNA replication. Tons of DNA damage, cells die. And some examples of this that we use today, topotecan, doxorubicin, etoposide. After that, we kind of move into this new era, the molecular era. Okay, so it represents a pretty big shift in kind of the approach that we're taking in chemotherapy. And this era has been enabled by this exponential increase in our molecular understanding of what's going on in cancer. And in some areas, this has delivered really miraculous results. But surprisingly, in a lot of areas, despite our deep knowledge now of cancer molecular biology, has not worked really as well, I think, as we expected or hoped. But targeted therapy, immunotherapies, targeted therapies often focus on inhibiting the driving mutation of a cancer, and immunotherapies are about harnessing the immune system to attack cancer. But I want to focus on just these first 50 years of chemotherapy, because I think that it highlights an important vulnerability of cancer. So when you look at all of these agents, alkylating agents, the antimetabolites, antimitotics, DNA crosslinkers, and topoisomerase inhibitors, they all just basically do one thing. And that is that they just indiscriminately kill dividing cells. And this makes sense that it would work against cancer, because cancer is a disease of uncontrolled cellular proliferation. But the fact that this works at all is pretty surprising, okay? Because our body is full of dividing cells. Our GI tract, is constantly recycling itself. Tons of cell division, the cells in our bone marrow, the cells in our skin, which is often why with these chemotherapy drugs, you see toxicities, because they just kill all dividing cells. Obviously, toxicity is a big problem. But again, I think the fact that this works at all, that there's a dose of these poisons that you can give, and it will kill cancer cells without completely demolishing your GI tract, highlights there is some special vulnerability here, that cancer is uniquely vulnerable to being attacked at kind of the focus of cell division. And so, I think chemotherapy, it packs this punch. The fact that despite all the advances we've made, often these chemotherapies are still the first-line treatment for a lot of cancers, highlights that there is this power, this explosive power of chemotherapy, of attacking cell division. But obviously, ideally, what you would want is some way to target that, to focus that so that you could kill specifically the cancer cell while sparing, the normal cells around it. So this is the kind of the question that we approach this with, is can we design a therapy that exploits the cell division vulnerability of cancer, that specifically targets tumor cells, but spares the normal cells in our body? And so in order to explain how we did that, I want to talk about chromosomes, okay? So this is chromosome. And I want to talk about this phenomenon called whole genome doubling. So whole genome doubling is a thing that can happen when a cell divides. So this, is a diploid cell. All the cells in our body are basically diploid, right? And what it means is you have 2 copies of each chromosome, one from mom, one from dad, and that's true for all 23 pairs of chromosomes. When a diploid cell is about to divide, it goes through S phase, right? It replicates all of its DNA and emerges from S phase in G2. And now, instead of having 2 copies of each chromosome, the cell, briefly in its life, will have 4 copies of each chromosome. It then lines those chromosomes up during mitosis, pulls them apart, and then ultimately separates into 2 daughter cells. And this final step in mitosis is cytokinesis, when the cell actually separates into the 2 daughter cells. 2 daughter cells emerge that are diploid. They're perfect clones of this parent cell. But sometimes, you could imagine this is happening in us billions of times a day. But sometimes this process goes wrong. And when it does go wrong, at this final step, the cell can fail cytokinesis. So it's gone through the whole thing. It's doubled all its DNA and now has 4 copies of each chromosome. But instead of dividing into 2 cells, it just kind of snaps back into a single cell. But now it's a single cell that has 4 copies of each chromosome. So this is a tetraploid cell. So this is the phenomenon of whole genome doubling. For the most part, this is not a big deal. If you have a tetraploid cell in your body, it can just perform its function, live out its days, and die, and it won't really cause a problem. But if this tetraploid cell tries to divide again, that's when the problems can happen. So you can imagine now you have this tetraploid cell, and it starts to go through the process of division again. So now it'll go through S phase. It makes a copy of all its DNA. Now it has 8 copies of each chromosome. It tries to line those up during mitosis. It separates them. It has a hard time separating them because now there are 8 copies of each chromosome in the cell. The machinery is not built to properly separate all of these chromosomes. And so what happens is you get a lot of chromosomal segregation errors in the daughter cells that emerge. Maybe sense too many one way, not enough the other way. The chromosomes break in half. And so what can emerge are these daughter cells that are what we call chromosomally unstable. And they end up looking, something like this. Okay, so on the left here is a karyotype, just the chromosomes of the normal diploid cell in our body. So that's what it should look like, right? 2 copies of each chromosome. But on the right, this is a cell taken from a breast tumor. And this is a whole genome doubled cell. So rather than having 2 copies of each chromosome, it has about 4. But you can see that there's some that has 7, some, half are broken off. And so this is characteristic of what a whole genome doubled cell looks like as it continues to divide and demonstrates this genomic and chromosomal instability. So in terms of whole genome doubling in cancer, there's some important connections here. So I'm going to talk about kind of 3 basic kind of foundational facts about whole genome doubling in cancer. So the first that I've already alluded to is that whole genome doubled cells display chromosomal instability, right? So with each division, there are gains and losses of whole or parts of chromosomes. And what this does is it allows cells to kind of, constantly be sampling different genetic permutations. And, a lot of these daughter cells that emerge, they're not going to be viable. The cells will just die. But if you are just unlucky enough, one of those daughter cells that emerges is going to have just the right combination of gains and losses to become a cancer cell. The other thing is that whole genome doubled cells, due to their additional chromosomes, are able to buffer against deleterious mutations. So you can imagine you already have this process going on where all these daughter cells are being made that are constantly, in a way, sampling all these different genetic permutations. DNA mutations are also happening. Mutations, can often be bad and lethal to a cell. So you can imagine, you're a normal diploid cell and you have 2 copies of a gene. And if you get a mutation in one of those genes, it would be lethal. The cell would die. But if you're a whole genome doubled cell, now you have 4 copies of these genes. So you can imagine you get a mutation that would otherwise be lethal to a diploid cell, but because you're a whole genome doubled cell and you have all these extra chromosomes, you can survive that mutation. So on the one hand, you're sampling a lot of different genetic permutations. And then on the other hand, you can buffer against all these deleterious mutations much better than a diploid cell. And so what this creates is almost this perfect kind of crucible for developing cancer. And this was first demonstrated by this guy, Takeshi Fujiwara. In the early 2000s, they showed that whole genome doubling on its own is sufficient to give rise to tumors. And so what they did with this experiment is they just took normal mouse mammary epithelial cells. They didn't do anything to transform them or make them cancer. All they did is they just made them whole genome doubled. And they took these whole genome doubled normal epithelial cells and injected them into mice, and they grew tumors. And so this showed that you don't need, whole genome doubled cells, they don't need any real extra help. They just need to be able to divide. And if you inject dividing whole genome doubled cells into mice, tumors will arise. So these kind of 3 facts, I think I just want to underscore how important these are in terms of this relationship between whole genome doubling and cancer. They have chromosomal instability, They can buffer against deleterious mutations. And because of this, whole genome doubling on its own is sufficient to give rise to cancer. And so from the perspective of cancer, whole genome doubling, it's like a very powerful adaptation. In a way, it's just like a one-way ticket to durable tumorigenesis. It portends a worse prognosis. If your cancer is whole genome doubled, it's more resistant to treatment, more likely to metastasize. And honestly, I think what this reminds me of in a way is this kind of old German myth. All right, this is the story of Faust. Okay, and this myth is like the story of the original deal with the devil. And what I think the moral of the story kind of underscores the risks of trade-offs and how something can seem very appealing, right, but it has like a fatal flaw within it. And in the spirit of bringing some poetry into the sometimes kind of sterile halls of medicine, I'm going to read a bit from the German poet Goethe, who, in his famous kind of telling of this story. So to set up the scene of what's being represented here, Faust is this guy on the right, and he's like this scholar who is disillusioned with the world. He feels like he's learned everything there is to learn. He's not finding any joy in life, and he's pretty just sad and depressed. And so this character, Mephistopheles, who represents the devil, comes to him and offers him a deal. And so he knocks on the door. And so Faust says, "A knock, come in. Again, my quiet broken." And then Mephistopheles says, "'Tis I. Come in. Thrice must the words be spoken. Come in then." And so Faust invites this devil in. And, he makes an offer. He says, to Faust, "In this sense, even canst thou venture, come bind thyself by prompt indenture, and thou mine arts with joy shalt see, what no man ever saw, I'll give to thee." And what Faust is looking for is he wants this moment of transcendence. And because he's so disillusioned with life, he thinks it will never happen. So he says, "I'll make the deal with you. If you can give me a moment of transcendence where I acknowledge, wow, this is, the greatest experience, then I'll die at that moment and you can take me down to hell." And so they make the deal. And, Mephistopheles, in true just classic villain fashion, after Faust leaves, he then he just says to nobody in particular, he says, "Dragged through the wildest life, will I enslave him through flat and stale indifference, with struggling, chilling, checking, so deprave him, that to his hot, insatiate sense the dream of drink shall mock, but never lave him. Refreshment shall his lips in vain implore, had he not made himself the devil's knock could save him still, were he lost forevermore." And I'm not going to tell you what happened, so you got to go read the poem, but I will hint that it doesn't end well, okay, Faust. And so, as I was thinking about whole genome doubling and cancer, I wondered, is this the same thing, right? Is this some evolutionary deal, right, that provides to cancer this real world of benefits, but also comes with some deadly cost? And so, first of all, I just want to talk about, so I guess the question then is, is whole genome doubling a Faustian bargain for cancer cells? And because I'm giving the talk, I will tell you, yeah, whole genome doubling is a Faustian. The answer is not no, it is a Faustian bargain for cancer cells. But I want to talk about now just how common whole genome doubling is, because even if this is some fatal flaw, if this only affects a really small number of cancers, I think it's less relevant. So we first set out to answer the question, just how common is whole genome doubling in cancer? And so in order to answer that question, we looked across 10,000 tumors. There's this database of tumors called the Cancer Genome Atlas. And what we did is written all along the bottom here are all these different tumor types. And each of these dots represents a sample from the database. And we just characterized and said, how common is whole genome doubling? And so if I zoom in just on a single type here, if we look, for example, at ovarian cancer. So all of these kind of dark blue dots at the bottom, those are not whole genome doubled ovarian cancers. But everything above it, the light blue and the black, those are tumors that are whole genome doubled. And so if you look at all of the samples in this database, 56% of ovarian cancers are whole genome doubled. And always, when I talk about this, I get this question, so I'm going to answer it now, is that the question is always, well, tumors they have a lot of heterogeneity. And so when you're talking about whole genome doubling, is it Is it some part of some fraction of the tumor that's whole genome doubled? And the answer is that whole genome doubling is this very early clonal event in tumor development. And so when we're saying that 56% of ovarian cancers are whole genome doubled, what we're saying is that, just over half of ovarian cancers, all of the cells in those tumors are whole genome doubled. It's not just some fraction. But when we looked at how many cells, it's often like 99% or something. So whole genome doubling is this clonal event that's describing the cells in the entire tumor. So you're either a whole genome double tumor or you're not. So when we looked across, this entire database and said, okay, across all of these solid tumors, how common is it? The answer is 36%. So over a third of solid tumors are whole genome doubled. So whole genome doubling, right? It's this potent tumorigenic force. You can think of it as like this dam. It's harnessing this power of all these extra chromosomes. And it's broadly shared across tumor types. And so the question, that we asked is, can it be exploited? Is there some weakness with this that we could take advantage of? So in order to answer that question, we turned to another database. This is called DepMap. This was this undertaking called Project Achilles by the Broad Institute. This is just remarkable what they did. Okay, so we have all these cell lines that you can use in research that ultimately came from tumors and then were taken and then cultured and now you can use to research a specific tumor type. So what they did is, they took a specific cell line. So, for example, they took a colon cancer cell line. And with this cell line, they wanted to answer the question, how important is a given gene to the viability of this cell line? And so using RNA interference, they would knock down the expression of that gene and then just ask the question, what happened to the cells? Did the cells die? If the cells died, you would say, okay, this gene is really essential to the survival of these cells. And they gave it a score saying, okay, essential. At the same time, if you took a different gene, you knock that gene down, and the cells don't mind at all, they just proliferate normally, you say, okay, this gene gets a different score. It's not essential at all. And so they, went through gene X, they knocked it down and then got a score, said, how essential is this gene? Then they moved on to the next gene. And then they did that with every single gene in the genome, 30,000 genes. They went through one by one and gave each gene a score. So then, for this particular cell line, this colon cancer model, you could look up any gene you want and say, how essential is this gene to the survival of these colon cancer cells? Okay, so they did that. And then they did that with hundreds of cell lines. Right? And then they did the same thing again with CRISPR. And they compared both, using RNA interference and CRISPR. And they came up with this, remarkable database that you can search and look up the essentiality of any gene. So this is, kind of what the data would look like. If you some gene you're interested in, gene X, and all of these dots represent different cell lines within these different tumor types. And you could look and say, oh, look, this gene, for whatever reason, in the cell lines over here is really important, and in the cell lines over here, this gene is not. So what we did is then we looked at the whole genome doubling status of all the cell lines, so that then we could map that onto their data. And so then it would look something like this. We could separate out the cells and say, oh, look, all of these pink cell lines, these are whole genome doubled cancers. And these gray ones, these are not whole genome doubled cancers. And we were interested in genes that looked like this, right? You can see that most of the whole genome doubled cells, for whatever reason, this gene is like essential if you're a whole genome doubled cell. But if you're not, if you're one of these gray cell lines from one of these non-whole genome doubled cancers, this gene is not really important. And we came up with this scoring system that is not really important, but basically we got this list of genes, and the higher the score of the gene, it just meant that that gene was more essential in a lot of different tumor types. So it wasn't just some specific thing to colon cancer, but if this gene, it was really essential in whole genome doubled colon cancer and breast cancer and ovarian cancer, we would get a higher score. And when we looked at what are the function of these genes, it's things like the spindle assembly checkpoint. This is just an important part of mitosis, or just kind of generically mitosis in general or DNA replication. And so, what that told us is that definitely whole genome doubling, as you might expect, requires genetic adaptations. So if you're a whole genome doubled cell, suddenly mitosis becomes more important. These genes that are helping mitosis happen become crucial to the survival of the cell. We then looked at gene expression. So we compared the gene expression of all whole genome double tumors compared to non-whole genome double tumors. And so, looking at this graph, on the right, the dots kind of arching up and to the right, these represent genes that are overexpressed on whole genome double cancers. We mapped this scoring data to see how they were expressed. And so we were most interested, right, in this kind of top right corner. So these are genes that are become really essential if you're a whole genome doubled cell. And these are genes that whole genome double tumors tend to turn up. They overexpress them. And there's one gene in particular that we were really interested in. So if you zoom in here, so KIT18A, this is the gene that really caught our attention. Because these other genes around it, these genes are like crucial genes for any cell to survive. If you get rid of, say, MAD2, this gene MAD2L1, it will kill all cells. You can't go through a normal mitosis. Whether your whole genome doubled or not, you need this gene. But KIT18A was interesting to us because we had never heard of this gene at all. This is like a very kind of relatively obscure gene. Not a lot of work had been done on it. And so we looked into what is this KIT18A that's scoring high on our genetic scoring thing. It's overexpressed, but we really don't know anything about it. And so we looked into it, we found out it's one of these kinesin proteins, okay? So these proteins, they use ATP, and they can walk and move up and down microtubules. And so KIT18A helps with chromosome alignment during mitosis. It walks up the microtubules of the mitotic spindle and just helps the microtubule dynamics line up the chromosomes really nicely during mitosis. So here's a video I made of a cell. So this is just the nucleus of the cell in white. The DNA is stained in white here. And what I'm going to show you is this cell's going to go through mitosis. So you see the chromosomes, they're going to condense, they're going to line up really nicely, and then they separate into two daughter cells. So this is just this normal, non-whole genome doubled cell going through a normal mitosis. If you take this cell and you get rid of KIT18A in it, you'll see it's going to go through basically the same thing. But if you watch closely, you'll see that the chromosomes, they don't align quite as well. There's a little more of this kind of chaotic dance as the chromosomes are trying to line up, but then ultimately it separates. It's pretty subtle. And in fact, that has no effect on the mitosis of the cell. It goes through a normal mitosis. There's no, any sort of damage done to the cell by this. These daughter cells will continue to proliferate as normal. And so this is the crucial thing, is that KIT18A is a non-essential gene in normal cells. Diploid cells that have a normal chromosome number, they don't need KIT18A. And this is best evidenced by the fact that there's this KIT18A knockout mouse that exists that is totally normal, has a normal lifespan, it's not prone to any other kind of diseases. So this mouse from fetal development through normal life, not a single cell ever sees any KIT18A. It doesn't seem to really have any deleterious effects. So KIT18A, basically useless in a normal cell. It does not matter if you have it or not. And that makes it a very exciting target for some reason, whole genome doubled cells do need this gene. So in order to validate this finding that we had found using all these computational efforts, we took 3 different cell line models, okay? So there's RPEs, these are just a normal epithelial cell line. MCF10A, this is a normal breast epithelial cell line. And HCT116, this is a colon cancer cell line. All of these are diploid, okay? They just have they're perfectly diploid. They have 2 copies of each chromosome. And what we did is we made what are called isogenic pairs of tetraploid versions of these cells. So I told you that if a cell experiences a whole genome doubling and then it starts to divide, it has all this chromosomal instability, you get these gains and losses, but not in these cells. These cells are like specifically engineered and selected for so that they're just tetraploid and they'll continue to divide perfectly stable. The daughter cells will just keep making tetraploid cells. And so the only difference between the diploid and tetraploid versions of these cells are just that one has 2 copies of each chromosome, one has 4 copies of each chromosome. But other than that, they're genetically identical. So we made diploid and tetraploid versions of these cells. And then we got rid of KIF18A. And so if you look, just starting from the left, if you look at the RPE cells, these normal epithelial cells, 2N is representing the diploid cells. You can see that when we have, normal amounts of KIF18A, and then we get rid of KIF18A, just a very minimal effect. Doesn't really have any, noticeable effect on viability. But for the whole genome doubled cell, the one that has 4 copies of each chromosome, this significant decrease in viability is associated with loss of KIF18A. And that was true in both the non-cancer cell lines and also HCT116, this colon cancer cell line. That when you experience a whole genome doubling, even if you're otherwise genetically identical, suddenly you become exquisitely sensitive to loss of KIF18A. So I'll show you again what this looks like. On the left, this is this diploid cell we already saw. You get rid of KIF18A, normal mitosis. But now if you're a tetraploid cell that's undergone a whole genome doubling, when you get rid of KIF18A, if you watch, it's trying to go through mitosis, it's trying to line up its chromosomes, but it can't do it. It gets stuck in mitosis. And it's, now spending hours and hours and hours in mitosis. And finally, this cell actually makes it through. It divides into 2 cells, and you can't really see well, I think, with the lighting here. But around those 2 nuclei, there are small little white spots. And what they are, those are chromosomes that have been separated from the main body of the nucleus. And so that's basically a death sentence to these cells. When they try to go to divide again, ultimately they'll die. And so if you're a whole genome doubled cell, you need KIF18A to successfully complete mitosis. So we then looked at a panel of breast cancer cells. I just had to show at least one Western blot during my presentation because I just did so many of these terrible things. And so these are 10 different breast cancer cell lines. So the cell lines on the right, highlighted in blue, these are whole genome doubled breast cancer cell lines, and the one on the left, these are not whole genome doubled breast cancer cell lines. And so now this is different, right? These are not cell lines that we have engineered and made to go through a whole genome doubling. These are just taken from patients, and that these 5 happened to be tumors that in their evolution underwent whole genome doubling, and these 5 here were ones that did not. And so if you look at the protein levels of KIF18A, you can see that if you're a whole genome doubled cell, you tend to have a lot higher level of protein of KIF18A, underscoring its importance. And when we got rid of KIF18A in these breast cancer cell lines, we expected a similar thing to happen, that they would depend on KIF18A for their survival if they were a whole genome double breast cancer cell line. And so here's an example of one of these whole genome doubled breast cancer cell lines. And this video is going to play basically over 3 days. It's just going to show the division of the cells. So on the left are the controls, the breast cancer cell lines that have their normal amount of KIF18A. And on the right, these are cells where we've depleted KIF18A. And so you can see the difference that on the left, these cells, they're just vibing. They're dividing, they're having a great time. And the cells on the right, it's the opposite. Right? These cells are having a hard time. And you can see that as time progresses, these cells are proliferating, and this just looks like, a battlefield. All of these cells die. So again, underscoring just the exquisite sensitivity that if you're a whole genome doubled cell, suddenly KIF18A becomes this crucial gene for your survival. So we quantified that data here, and you can see that, again, it's the blue cell, the whole genome double breast cancer cell lines that tend to have the big dips in viability. And so, this demonstrated across a range of breast cancer cell lines that loss of KIF18A, it selectively kills whole genome doubled cells. So I've tried to demonstrate that whole genome doubling, it's a potent driver of tumorigenesis, that about 1/3 of solid tumors are whole genome doubled. That whole genome doubling confers an exploitable sensitivity to loss of KIF18A, and the depletion of KIF18A kills whole genome doubled cancer cells across different tumor types in vitro. We showed it in colon cancer, we showed it in breast cancer. And so we were really excited about this. We filed this patent to try to patent this idea about targeting KIF18A, but the problem is we didn't have an inhibitor. We didn't have this small molecule inhibitor. It's just kind of the idea. This is a pretty hard patent to protect, but we just thought, well, let's just file this anyway. And the other, an important thing if you do research, that every day you go to work with this constant fear that you're going to open up some email and you're going to see that somebody published the work that you're doing, right? So I lived with that fear for a while. And eventually, even though we didn't have the inhibitor, we thought we just have to publish this. This is just too exciting. So we put this up on bioRxiv. This is open access. And so this was in June of 2020. And then one day I went to work and I found this patent application by a little company called Amgen. And they had patented 20 small molecule inhibitors to KIT18A. So in the time, so we've been looking for 6 months trying to find one, and then they found 20 relatively quickly. And then we were able to publish it in Nature. It was really exciting. And then they just very quickly moved into phase 1 trials with their drug. And then this thing really took off because it got fast track designation with from the FDA. Another company was started called Volastra that was just based on basically targeting KIT18A. And they ended up licensing this Amgen's drug, so it's called Savilnasib. And right now that's in phase 1 and 2 trials. And then Volastra got $1 billion from Bristol Myers. And so, when I go to bed at night, sometimes, and I think about, especially on a hard day, maybe I'm feeling down, it just warms my heart to know that somebody got $1 billion from this, even though it was not me. And now, more companies have joined in trying to find inhibitors to treat, to target KIT18A. And then Volastra, they started again their own phase 1-2 trials and kind of a basket trial of lots of different tumor types for a different KIT18A inhibitor that they also developed. But to end, I just want to share, Amgen finally published the paper on their inhibitor at the very end of last year. And so they use, they inhibit KIT18A to target these chromosomally unstable cancer cells, which is, in a way, basically synonymous with whole genome doubled cancer cells. And so this is some of their data. So the first thing they look at is what effect does inhibition of KIT18A have on bone marrow cells? And so DMSO on the left, that's just the control. And then the first 4 things you see in each of the graphs, the AM dash, whatever, these are the KIT18A inhibitors. And so if you look, you see this, so the BRDU population, this is just looking at DNA replication. How well is DNA replicating? So you can see compared to the control, the KIT18A inhibitors, there's no difference. It doesn't affect DNA replication in human bone marrow cells at all. But when you compare that to the last 3, right, these 3 are kind of, you can think of them as common chemotherapy drugs that work by these more classic mechanisms. You can see that they significantly reduce DNA replication in the bone marrow cells. When you look to the next graph over to the right, the sub-G1 cells, it's looking at cell death. So again, the DMSO, the control, you compare that to the 4 KIT18A inhibitors, there's really no difference. It's not killing the human bone marrow cells, but then the next 3, these more common drugs, significant cell death. And then lastly, if you just look at you just count the cells and say, how well are these human bone marrow cells proliferating? Again, the control and the KIT18A inhibitors are no real difference. The more standard chemotherapies significantly impair bone marrow cell proliferation. So it showed that KIT18A inhibition is not toxic to human bone marrow cells in vitro. The next thing an interesting thing they did is looking at sensory neurospheres. So what you can do is you can take pluripotent stem cells, induce them into neurons, and then they grow in these spheres, and you basically just count the number of neurons there. So at the top, the DMSO control show what these sensory neurospheres look like. And here's what like paclitaxel and vincristine look like, right? So it's well known that one of the toxicities with these drugs are you get bad neuropathy. And sometimes it can be so bad that you have to stop treatment for these patients. And so you can see it just demolishes these neurospheres, specifically the vincristine, even a very low concentration, just wipes out the neurospheres. KIT18A inhibition, even at a concentration 10,000 times higher than what the vincristine is, really no effect on the neurospheres. And so, this demonstrates that KIT18A inhibition is not toxic to human neurons in vitro. And then some of their animal model data. So they use an ovarian cancer mouse model. And I think that this is really underscoring the broad applicability of this, right? We showed it in breast cancer and in colon cancer, and in their model, they use ovarian cancer. And so what they do is they inject these ovarian cancer tumor cells into mice. The black line just shows how the tumor grows over time. And the blue line represents docetaxel. It's one of these anti-mitotic chemotherapies. And you can see that the docetaxel, it limits the growth of the tumor cell, which is great. But compared to the KIP18A inhibitors in red and green, they completely demolish this tumor and shrink it basically down to nothing. And then when you look at safety and tolerability in the mice, so this first graph is just showing body weight of the mice, you can see that the black line again is control, that the control mice, they tend to gain a little bit of weight through the experiment. And the blue line, the docetaxel, maybe they're more just kind of keeping their weight even. Whereas again, with the KIP18 inhibitors, you're seeing they're actually gaining weight during the experiment. And then I think this is, one of the most significant things is you look at neutrophil count. So you're looking at neutropenia. And you can see that, again, the control compared to the red and green show real no difference. These mice are not getting neutropenia. But, you treat them with docetaxel and you see the significant reduction in neutrophils. And so I hope that I've demonstrated that the efficacy of chemotherapy, it shows that cancer cells are particularly vulnerable to attacks on cell division. There's some weakness, some magic there that grants us this power to be able to target them in a really powerful way. And I've showed that whole genome doubling confers unique advantages to cancer cells, but there's also this fatal vulnerability that comes with it, making it truly a Faustian bargain to experience a whole genome doubling. And I've showed that by inhibiting KIP18A, you can specifically attack dividing cancer cells and spare the normal cells that make up the rest of our tissue. And so I just want to also end with some acknowledgements before I take questions. So this was my PI, Neil Ganem, at BU. Neil is just this brilliant scientist and amazing person. And one of the luckiest things that ever happened to me was that I got to work in his lab and be a part of this project. So many people helped and could not have, come close to doing any of this without the help of these people. And also, as Dr. Wittelis just mentioned, I just finished residency 2 days ago. And so I just want to acknowledge the residents that are here and that are listening, because honestly, residency is this really hard thing. I would say we're right up there with one of the hardest things that I've done. And something that is like not hard at all is working with these, amazing people. I think it really has been like an honor of a lifetime to just work with people here who are just so smart and brilliant and so kind. And, I'm forever grateful for the opportunity that I had to work with all of you. I also just want to briefly acknowledge my wife. Having a baby during residency, also super hard. And I could never, have done any of these things without her support. She's such an amazing mother to our baby Everett, who just turned 1. And then lastly, I just want to show you, this is a picture of the town that I grew up in. I took this picture from the hill. This is a whole town, okay? And so I grew up in just this tiny remote place in the middle of nowhere in Canada, kind of where you can see the Rocky Mountains on the horizon. And I think the fact that I tell you, I'm the only person from this town to ever stand here. That's a guarantee, because I know everybody from this town. And I think just the fact that I am here, I think, is just a real testament to my parents, who I think just raised me with this, instilled in me this belief that I could do anything. And I'm forever grateful to them for that. So thank you.
All right.
Okay. I don't think anybody was disappointed. That was spectacular. So I imagine there may be some questions. Thanks
for a great talk. It's Ashley Visitor from Oncology. Can you tell us, I have a couple questions. Can you tell us whether this principle that you showed for whole genome doubling susceptibility to inhibitors like the KIT that you mentioned applies also in almost whole genome doubling hyperdiploidy, particularly for lymphocytes and leukemia and myeloma?
Yeah.
And then the second question is, how do we reconcile these in vitro observations to what we see in vivo where hyperdiploid cancers, some of them that have EBV don't seem to be affected by, whole genome double cells don't seem to be affected by microtubule inhibitors the same way as you might imagine.
Yeah. So I think that, the question, it highlights really the core question I think that we still have is what are the biomarkers or what are the features that actually predict whether a cell is going to respond to KIT18A inhibition or not? So, I tried to argue that whole genome doubling confers the sensitivity to KIT18A. But, if you looked closely, I think obviously, as you alluded to, some of the whole genome doubled cells were not that sensitive. And so I think we don't have a perfect biomarker that will say, this is what makes a cell vulnerable to loss of KIT18A. And so I think that that's kind of what I think a lot of people are working on that question. So what the Amgen paper shows is that it's really about chromosomal instability. That even if you're not whole genome doubled, but you have some of these defects that are causing chromosomal instability, it makes you vulnerable to KIT18A. But also that's not perfect because there are some chromosomally unstable cells that don't respond to inhibition. There was a recent paper that came out that showed it has to do with vulnerability of the spindle assembly checkpoint. So the spindle assembly checkpoint is this kind of function of the cell where the cell is going to line up all its chromosomes in mitosis. And you have to satisfy this checkpoint. You have to get everything lined up right. And once the checkpoint is satisfied, then the cell can proceed with mitosis. And so there was data that showed cells that have weaknesses or vulnerabilities at the spindle assembly checkpoint, that is actually the trait that is defining KIT18A sensitivity. And so I think it's an ongoing question. So I think, like a lot of things, we don't have a perfect answer. In broad strokes, we say in general, whole genome doubled cells or in general, chromosomally unstable cells. But I mean, that is the crux of it, is finding the actual biomarker and the true mechanism says this is what makes you sensitive. I think is still an ongoing question.
So thank you. This is Anne Malani from Hematology. Phenomenal talk. Thank you so much. So I have a question around the whole genome doubling sufficiency to cause cancer. So I noticed in the people you referenced from David Coleman that they used P53 null memory cells. Yeah. So my question is, in the absence of P53 null, is it still sufficient? And also, does related to that, does the sensitivity to the KIT18A inhibitor, is that independent of P53 mutational status? So for example, if the cell is P53 null or mutated, is there still sensitivity to KIT18?
Yeah. So in the Amgen paper, they looked at what mutations correlate most strongly with KIT18A sensitivity. And unsurprisingly, it's P53 mutations. So the reason in that paper that they used P53 null mouse memory epithelial cells is because if you have intact P53, whole genome double cells will not divide. The key thing to get a whole genome doubling cell to divide is inactivation of P53. And so it almost comes part and parcel with whole genome doubling because you need some way to get around P53 in order for this whole genome doubled cell to start its proliferation in general. And so I think the general answer to your question is you really need a P53 mutation to be sensitive if you're a whole genome doubled cell. So yeah.
Time for one final question, though, please.
Oh, only one I want to ask you. That's fabulous talk. A more general biology question, and then I'm just amazed at this Herculean effort at the Broad, but you showed a mouse that is completely fine without this gene. How many genes in our genome or mouse's genome are not needed? Second question, Faustian bargain. What's our purpose at a university in terms of learning and knowledge versus filing patents and making billions of dollars? aren't all the people that are suppressing publication of their data until they can lock down IP, in fact, making a Faustian bargain?
Wow, questions and up.
Dr. Kalidis. All right. The answer to the first question is 5,974 genes. I just made that up. But it's, I think the answer is a lot. I think that there are a lot of genes, that we got 30,000 of them, and a lot of them, have evolved over time, that are kind of baggage, I think, carried along through evolutionary history. And there's a lot of redundancy in a lot of their functions. So I don't know the exact number, but I would suspect it's not rare. I suspect there's a decent number of genes that you can get rid of and would probably just be fine, although I don't know the answer. And to your second point, yes, Dr. Kalidis, that is the true Faustian bargain, which is why I sleep so well at night. I'll publish this data. Who cares if someone else gets it?
All right, and that, let's give a round of applause.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.